Welcome to our stop motion video on the sliding filament theory. In the next minute, we're breaking down how your muscles actually contract. We'll walk through EC coupling, the cross-bridge cycle, and how actin and myosin slide past each other to create movement. Before EC coupling begins, the signal has to reach the muscle. Here at the neuromuscular junction, a motor neuron meets the muscle fiber. When the nerve impulse reaches the end of the neuron, it releases ACH into the space between them. ACH then binds to receptors on the sarcolemma, and that binding starts an action potential in the muscle fiber. Next, we have EC coupling, and that starts with the spread of action potential. The action potential now spreads quickly along the sarcolemma. As it travels, it dives down into these T-tubules, carrying the electrical signal deep inside the muscle fiber, so every part of the muscle gets the message to contract. When the action potential reaches the sarcoplasmic reticulum, it forces the calcium channels to open. Calcium floods out of the sarcomere, and that release is what unlocks the next stage of contraction. Calcium immediately binds to troponin, causing it to shift tropomyosin out of the way. This exposes the binding sites on actin, allowing myosin to finally grab on.
With the binding sites on actin now exposed, the energized myosin heads attach to actin, forming what's called a cross bridge. This connection marks the very start of the muscle contraction. Once the cross bridge is formed, the myosin head pivots in a powerful movement known as the power stroke. This pulls the actin filament towards the center of the sarcomere, causing the muscle to shorten. To detach the myosin head from actin, a new ATP molecule binds to the myosin. This causes the cross bridge to break, allowing myosin to release the actin filament. The ATP molecule is then split into ADP and phosphate. This hydrolysis recocks the myosin head into its high energy position, ready to form another cross bridge as long as calcium stays bound to troponin.
With the cross-bridge cycle repeating over and over, the actin filaments slide inward toward the center of the sarcomere. As both sides pull in, the entire sarcomere shortens. This is how muscle contraction happens.
When the nerve signal stops, calcium is pumped back into the sarcoplasmic reticulum. Without calcium, the binding sites on actin close, cross bridges stop forming, and the muscle returns to its resting length.